Hello students. So welcome to the exam for review. We will discuss about some of the important points, but as you know, and I have told you last time also during the exam 3 review, to me, every single slide is important. Every single detail is important for memorizing purpose as well as understanding purpose. So for me, actually review means pretty much going over the whole thing, but briefly. Okay, so let's start this. So we'll start from here, lecture 7, because until lecture 6 we covered earlier. Uh, this is just the title. So about the filament assembly, that's the actin filament assembly. We discussed about FH2 domain and then rocking, and then profilin ATPG actin binding to the FH2 domain. It's a dimer. Formin is the one. FH means formin homology domain. So formin protein is the one which helps in the synthesis of long filament, and ARP23 is the one which helps in the synthesis of branched network. FH1 is proline rich, binds profilin, not profilin, but profilin, no G, ATPG actin, and FH2 binds profilin plus ATPG actin at plus end. Okay, here we discussed about the inactive state, that means bend state or folded state, where RBD is bound or nearby FH1 FH2 domain. When this comes in contact with activated Rho protein, which is Rho GTP version, this opens up and FH1 domain recruits the profilin G actin and then transfers them to FH2 domains. Excuse me. Near the FH2 domain, that's the plus end, and the growing end is the minus end. I mean, this is the growing end, so this is the plus end. The already formed unit will be the minus end at the end. For the ARP23, it's a 70-degree angle for the branched chain formation, and the starting point will be the negative end, and the growing end will be the positive end. Here, for the NPF, nucleation promoting factor, we have WCA domain, where it denotes WH2 connector and acidic domain. In terms of questions, it becomes important that which binds what. So for example, actin subunits gets bound by the WH2 domain, and the acidic domain binds the ARP23 complex. Okay. Um, probably will be important for filling the blanks or true false. Cytochalasin D, lateran choline, jasperquinolide, and phalloidin, these four names and what they actually do. These two promotes depolymerization, these two promotes polymerization, and through what kind of activity. So you'll need to know all these. Also that even at lower CC, since these two promotes polymerization, even at lower CC, actin won't depolymerize. The end result is eventual death for the cell. In that case, through ARP23, similar to the FH12 domain, WASP is one example of NPF. There are three different examples, WASP, WAVE, and another one. So for WASP, which is Wiskott-Aldrich syndrome, it's an X-linked disease. The RBD domain is close to WCA domain. This is at N terminus, this is at C terminus. This one has to open up and bind two different signals on the membrane. So this situation is called coincidence detection, because two input signals are present instead of one like previous. So this B domain binds to, or the basic domain binds to the regulatory phospholipid PI45P2. You don't need to know the full form. and the RBD domain binds to CDC42 in this case, obviously activated version, which is GTP. Okay. In this case, this is just an example to show how this works. I could have made question in a different situation, in a regular situation out of this, but this is just to show how the engulfing happens and how the actin filaments gets arranged around a groove, or rather invagination, during the phagocytosis. In this one, several things are important, yes, in terms of information. For example, fimbrine is present in microvilli filoporia and focal adhesions. Alpha actinin are present during in stress fibers filopodia and muscle Z-line. In cell cortex, we have stream, which forms a spoke and hub-like structure in RBC plasma membrane. Filaments are present in leading edge stress fibers filopodia. Dystrophin are present near the muscles, which links membrane proteins to actin cortex in the muscle. Okay. So filamin also forms a loose 3D network with gel-like properties, which is also mentioned here, all the details. So here there are two steps through which we can get actually the S1 head out of this whole uh, myosin structure. So this is myosin 2 structure. First, digestion with chymotrypsin will give us HMM and LMM. The second digestion through papain will give us S1 head and S2 head. And S1 head is the important in terms of studying in vitro mechanism. So this type 2 of myosin has two sets of light chains, regulatory light chains and essential light chains. The cleft or the alpha helical neck, also known as C-clamp, passes through this essential side chain, and there is a nucleotide binding site on one end and actin binding site on the other, on the S1 head portion. Excuse me. Okay, these two light chains act as levers during the myosin force production. That's also important information. Here, this experiment just shows how it moves, that these are plus end directed motor. So the S1 heads actually reach, although they are fixed on the cover slip, they actually reach towards the plus end, and eventually the actin filament moves towards the minus end. Here it's important because there are different or common classes of myosins rather. So the classes differs in terms of their variability in light chains. Here it's variable in two, there are two pairs. Here it's single type. Okay. And also 5 helps in organelle transport, 2 helps in contraction, and class 1 helps in membrane association and endocytosis. This is a very, very important slide in terms of understanding power stroke and what happens in different case scenario when there is no ATP versus ATP bound situation versus ADP plus PI bound situation, when exactly the cocked state happens in this situation, when exactly the power stroke happens, and power stroke is due to the what, like elastic energy. When there is no ATP, then what happens? S1 head release. So ADP release, ATP bound, S1 head release. These two happen simultaneously pretty much. Length of the neck increases. If it increases, it determines the rate of movement, because as it increases, the length of the neck domain, the rate of movement will also increase. In case of rigor mortis, what happens? For a long duration, there is no ATP. In this situation, this is no ATP and temporary binding. But in case of long duration of no ATP, it is permanent binding. So muscles get stuck. So they becomes hard. They do not move at all. Okay. This is just to give you an idea about the muscle fibers and the different zones. So H zone and A band. Know the difference between H zone and A band. During the contraction, actually H zone is getting contracted. Everything else remains same, pretty much. And also Z discs are coming close together. So that means this one sarcomere unit is contracting. The titin is basically this elastic protein here, and it binds between the Z disc and the M band. That's the location of titin. Nebulin is the one which binds the actin subunits, this black one going around. And nebulin also determines the length of the actin thin filament. How long is it going to be? Also, because the length is also determined by the capping of cap C at the plus end, so it doesn't let it grow further. So both of them work together to keep the length of the actin filament at a certain uh, length. This is important again because calcium, basically the sarcoplasmic reticulum, this is the reservoir for calcium, and when the electrical impulse arise from the motor neurons, the impulses travels through these T tubules, which causes release of calcium, and the calcium in the surrounding increases. And as the calcium in the surround increases, the myosin binding site exposed, causing contraction. As the calcium gets into the sarcoplasmic reticulum, so there is less calcium outside, less concentration, I mean, lower concentration of calcium. And also it's important that this is more than 10 to the power minus 6. Remember, we are talking about minus. So more than 10 to the power minus 6, it can be 10 to the power minus 6 or 10 to the power minus 5 or minus 4, anything above. This is less than 10 to the power minus 7. So 10 to the power minus 7 or less, basically. So less calcium outside environment will cause relaxation because myosin binding sites are masked. How are they masked? Because of the arrangement of these tropomyosins. Okay. This is little complex because a lot of names are involved, a lot of items are involved, but we need to know the flow chart. So I basically simplified this whole structure here in terms of flow chart. So myosin light chain basically gets the regulatory light chain basically gets phosphorylated, and then this myosin, basically this MLC phosphorylated MLC activates the myosin 2S1 head to myosin 2S1 head with ATPase, which is active version. This is not active but active version. There is one more mechanism also. This is more than 10 to the power minus 6. Okay, so calmodulin becomes calmodulin calcium. Okay, so there is another way, which is calcium independent way, where rho kinase basically decreases the myosin light chain phosphatase. So basically this decreases the production of this. And remember, this is involved in this step. So basically it will keep it inactive in this case. The myosin 5, which is involved with the cargo movement. So this structure basically shows that it is folded and inactive state, which is most of the cases for molecules. When it is bound to the cargo receptor, it is active and open state. Okay. So for freshwater alga Nitella, the movement in the cytoplasm happens because of the movement of these endoplasmic reticulum structures on top of the non-moving cortical cytoplasm through the myosin 5 activity, because myosin 5 is responsible for the cargo movement. In this case, the cargo is the endoplasmic reticulum itself. So due to the movement of these big molecules, or big organelles, not molecules, big organelles, the whole of the cytoplasm gets steered and they start moving. Okay, steps are important. So what happens in different case scenario? This extension is ERP23 dependent mechanism. That means a lot of branched chain network should form for the actin filaments. This one, adhesion, involves integrin mediated connection. Translocation based on actin myosin 2 dependent contraction at the rear of the cell. De-adhesion and endocytic recycling. Internalization of membrane and integrins at the rear of the cell. Okay, that's pretty much explained here. This is just to show you what happens in different stages or different portions of the motile cell. This is the leading edge. These are the stress fiber formation in between, and towards the end, integrins actually bound to the these actin filaments. Here, it's a general representation of activation of Rho proteins, which is a signal protein on the membrane. So activated version of Rho protein has Rho GTP in it instead of Rho GDP, and that happens due to the extracellular signal binding to the receptor on the plasma membrane, and that causes this Jeff activity, which causes replacement of GDP with that of GTP, and that further affects the effector proteins. And in the inactive version, we have a Rho GDP version along with GDI. Okay, GDI is guanine nucleotide dissociation inhibitor. So all of them are bound in complex form. So different Rho proteins are shown here. Basically, CDC42, RAC, as well as Rho, all of them falls under a Rho family of proteins. Okay. So CDC42 downstream, they decides polarity through affecting the PAR6 molecule. We haven't studied that, but we have to know this because this is important. Also, WASP affects the ARP23 through actin polymerization and philopedia formation. RAC affects the wave pathway, affects the actin to ARP23, causes the actin polymerization and lamellipodia formation. Rho, in fact, affects two different molecules. In one case, Rho kinase, which helps in the myosin activity, and formin, as we know, is involved with actin polymerization. Now, Rho kinase can inhibit or do inhibit myosin LC phosphatase, and this in general inhibit this. So since this inhibits myosin LC phosphatase or phosphorylation, presence of Rho kinase inhibits this activity, so they cannot further inhibit this. So this process can go on. So know about this chart. If required, make flow charts and practice. Also, this is important where I have shown different regions and how they are activated. So first, in the front, CDC42 helps with the philopodia formation, RAC, lamellipodia formation, both of them in the front, and then in the back, Rho helps with stress fiber formation. Now, if I say that on the back, Rho activation is affected by what? In general, it can be both of them, but in signaling pathway, we say the previous molecule or the next molecule. Okay, so Rho activation is affected directly by RAC activation. Yes, it does get affected by CDC42 as well, but that's much upstream. The immediate effector will be the rack. So CDC42 affects rack, rack affects Rho activation, Rho inhibits rack activation because they have to pull the back of the cell in the front. So that is why they have to stop this rack as Rho gets activated and moving forward, helps moving the cell material forward. Important in terms of multiple choice, that what kind of structures gets formed in case of different kinds of mutation. So dominant Rho active, they form abundant contractile stress fibers. Dominant CDC42 forms filoporia all around. Dominant active rack forms peripheral membrane ruffle. This one shows in terms of the effect of negative dominant negative mutation, and CDC42 dominant negative mutation wound closure still happens. The most it gets affected negatively is because of the dominant negative Rho mutation, because in this case there is very little wound closure or the movement, basically movement of the cell. So out of all these three individual mutation, the most the movement of the cell gets most affected by the dominant negative Rho mutation. Obviously, when they're happening individually, not together. Okay, the map proteins. Okay, the difference between microfilament, microtubule, and intermediate filaments. We did not discuss about intermediate filament because that will take a long time, and also, I mean, that is important, but in terms of our syllabus, we focused mainly on microfilaments and microtubules because a lot of material are there also to understand, also to memorize, also to know. So, but do know the differences, that these here actin binds ATP, here it binds GTP, here it doesn't bind any nucleotide. These two are highly dynamic, this one is less dynamic. These two are polarized, this is unpolarized. Here it creates a track for myosin, it creates a track for kinesin and dynein. There is no motor protein involved in intermediate filament. Okay. Also, their functions: contractile machinery and network at the cell cortex, organization and long-range transport of organelles, cell and tissue integrity. Okay. So the structure of tubulin dimers, we know that alpha and beta tubulin both are present, and together they're called tubulin dimers, where this GTP, which is bound to the alpha tubulin, this alpha tubulin GTP is non-hydrolyzable and non-exchangeable. This one, the GDP associated with the beta tubulin, is hydrolyzable and is exchangeable with GTP. Okay, there is another one, gamma tubulin, but we do not consider it because that is the starting material or involved with the microtubule assembly. Protofilament is one long filament is called protofilament. At seam, the arrangement is different versus other cases. So staggered arrangement versus non-staggered arrangement at the seam. At the seam, they are non-staggered arrangement, but otherwise they are staggered arrangement. So alpha and beta tubulin are close together side by side at seam, but other cases alpha is close to alpha, beta is close to beta, or side by side. Here we discussed about gamma Turk or tubulin ring complex, where gamma tubulin is involved. So alpha, beta, and gamma, here are the gamma tubulins at the end. So these red structures, these are gamma tubulin, but these are present only at the beginning, not at the end. So, okay. Important in terms of where they are found. Protofilament structure, where when there are 13 of protofilaments forming one single microtubule, in that case, it is found in cytoplasm, known as singlet. For doublet plus 10 arrangement, they are found in cilia and flagella. For triplet, they are found in basal bodies and centrioles. We have studied in detail about the cilia and flagella structure, as well as the centriole a little bit. Important about the arrangement, usually they are arranged because they have a polarity between the plus end and the minus end mTOQ center, but only in case of neurons they have mixed orientation, especially in case of dendrites. So dendrites can have plus end, minus end, and then minus end, plus end. The rest of the cases, rest of the examples, they have a single orientation. Okay. Here we discussed about the GTP version of beta tubulin assembly and disassembly here. So this process when it starts assembling is called rescue. This process when it starts disassembly is called catastrophe. Okay. Here, this is important because of the rates. The assembly is actually slower. That is why it takes more time. But the disassembly, it is faster, almost 7 times faster, so it happens very quickly. And then there is a rescue, and then again assembly happens, and then again catastrophe happens here. Disassembly, rescue, assembly, catastrophe, disassembly, then here, rescue, assembly, just like that. And it goes on as long as the cell needs more and more microtubule formation. So we discussed about the MAP proteins or the microtubule associated proteins. They bind at the C terminus or towards the plus end, and they also help in the stabilization or promote polymerization, and regulated by MAP phosphorylation by MARC, which is microtubule affinity regulating kinase. It can cause Alzheimer's disease and tau. We have discussed that in the chapter earlier lecture. Uh, plus tip proteins or plus end tracking protein activities or end tracking protein activities rather. So in this case, what is important is that other proteins plus tips actually hitchhike on EB1 proteins. So EB1 and XMAP 215, both of them basically promotes the microtubule growth or polymerization at the plus end. And clasps are the second type, which reduces the frequency of catastrophes, also in terms help in the polymerization. Here, two different molecules, kinesin 13 motor protein. This enhances the disassembly at the plus end or the minus end, and ATPase activity dissociates kinesin 13 from the alpha beta tubulin dimer. So if there is a ATPase activity, that means ATP hydrolysis. ATP hydrolysis produces enough energy to remove kinesin so that it cannot anymore disassemble at the plus or minus end. Okay. OP18 stathmin, they are important because they bound to two of the dimers, a pair of dimer. Focus on the pair of dimer versus a dimer. Okay. Also colchicine. Colchicine is an alkaloid derived from Colchicum autumnale. This inhibits mitosis by inhibiting microtubule polymerization. So this colchicine is a chemical which is basically um, depolymerization factor. Important point. Kinesin plus and directed motor slides antiparallel microtubules past each other, which is important. Antiparallel microtubules. Cytoplasmic dynein is a negative end motor and it acts along with dianactin complex and cargo adapter. This is to show which one moves among all these different examples, which one moves the fastest, which one moves the slowest. Vesicle moves the fastest, tubulin and neurofilaments move the slowest. Same here, folded state, inactive, active state here when it is open version. And in this case, although it is active, it's not moving because The ATP is non-hydrolyzable ATP that is used here to lock the organelle onto the microtubule. Just walking around. Okay, different types and differences. Kinesin 1 is the conventional one. So out of all these classes, kinesin 1 is the conventional one. Kinesin 2 is heterotrimeric. That's its characteristic feature. Kinesin 5 is bipolar and also involves during cell division, if you remember, because they bind to anti-parallel microtubules. This is minus n plus n, this is minus n plus n, and help pass slide each other. Kinesin 3 is a different thing. This is not a motor protein, rather this kind of helps in the depolymerization at the plus as well as minus end. Or rather it's a motor protein, but its function is a little different compared to others. Steps are very important. What's happening at what point? Because here, when it binds to the ATP, which causes a conformational change in the linker domain, which causes the trailing end to swing forward and become the leading end or leading head. Excuse me. What happens when the leading head releases the ADP? And here is the leading head. The leading head linker is not bound to the leading head. It's open. While the trailing head linker is bound to the trailing head. That is one important point. Same concept. While folded, it's inactive. While open, it's active pretty much. Important here because several things are going on. First of all, this is dynamidine. This whole structure is called dynactin. It's a complex. So complex contains dynamidine as well as one single actin filament plus 7 different kind of ARP1s, which is capped by caps Z at the end. So the length doesn't increase much. Second arm, this is the first arm. The second arm is P150 glued, which binds this dynactin, the whole complex, to this microtubule. Okay, excuse me. Also, it binds to the dynein. So dynein here, this one is the dynein. Okay, this is the stalk. Here we can see dynein molecule in detail. There are 6 ATPases. Okay, 6 AAA ATPases. Where they are, the linker domains are, that is most important. The first one, in absence of nucleotide, the linker is straight. Between the first and the fifth AAA repeats, which is this is straight. In case of ATP bound state, the linker bends between second and third AAA repeats, the second and third. Okay. Excuse me. Only one out of 6 ATPases, AAA ATPases, only one, the first one, is involved with the ATP hydrolysis, or at least this much information is gathered from studies. Pre-stroke and post-stroke. This is pre-stroke between third and 5. This is post-stroke between 1 and 5. I mean second and third, 1 and 5. This is pretty much same with an extra information. What happens when the dynamidine is overexpressed? As the name suggests, with dynamite, this will explode and separate the units. 2 molecules involved, NUD-E and LIS1. LIS1 defects causes this disease, Miller-Dieter leukoencephaly. And the symptom is having a smooth brain, that is because of lack of cortical folds and grooves, and causes defects in mental development for the LIS1. And LIS1 actually interacts with the ATPase domain of dynein. This is the dynein. These are the ATP, AAA ATPase domains. This LIS1 binds to the AAA ATPase of the dynein. This is just to show that different kinds of motors helps in different movement towards different ends. So dynein, since cytoplasmic dynein is a negative end directed motor, so it goes to the negative end, which is the mTOC. Kinesin is a plus end directed motor, so it takes the cargo to the plus end, that is the extreme end. What happens when it reaches at the extreme end? The motor protein switches and it gets back to the minus end. This is a good example to show that dispersion of melanophores. So again, when there is low CMP, it affects the dispersion. That means all the melanophores are concentrated, aggregated melanosomes. And in case of high CMP in the surrounding, the melanophores are dispersed so that it can have a clearer patch, or not patch, clearer skin in frogs. Here they have darker patch. Okay. Okay, what happens in case of post-translational modification in case of alpha and beta tubulin dimers? So this is polyglutamylation and polyglycylation happens with a specific glutamate residue, but they are mutually exclusive, and this can happen on both alpha as well as beta tubulin. But in case of tyrosylation or acetylation, it happens only in case of alpha tubulin. That is important. So microtubule in the axons are stabilized by acetylation and destyrosylation, which encourage kinesin 1 binding. Okay. Cilia flagella, as we discussed that we have discussed this. Here we have discussed the arrangements, and there are 3 different regions, and they are arranged differently. So at the negative end, this portion, this is called basal body, this underneath the plasma membrane. So this is basal body where the arrangement is 9 into 3 plus 1. So triplets plus 1 singlet in the center. 9 triplets outside on the periphery connected to 1 singlet at the center. In the transitional zone, they have 9 doublets, no singlet in the middle. Okay. Before that, it becomes 9 triplet and no singlet. Then slowly it becomes 9 doublet with no singlet. But towards the end, that is the axon, the longer portion outside the plasma membrane, here in the axoneme, the arrangement is 9 doublets with 1 single doublet here. So here we can see the structure that is present in axoneme, 9 doublets, and in the center there are singlet, 2 of the singlet, not doublet, sorry, my bad, 2 of the singlets, okay, and different proteins which are involved. Nexin obviously is involved with the A tubule versus B tubule, and as we know, A tubule is the one where there are 13 protofilaments, B tubule is the secondary one which has got 10. So A tubule and B tubule of the other gets connected by Nexin linker. Between these 2 singlets, they are connected by periodic ladder. And radial spokes are the one which helps them to not get collapsed, okay, inside, because of the presence of the radial spokes. So all of them can be the questions in true or false or multiple choice rather. Here we have seen how Nexin works. Obviously we need ATP, addition of ATP. So here we can see that dyneins are bound, And while the Nexin linker is present between the A tubule and the B tubule, in this case, movement or activity of dynein negative end actually moves these, like helps bend the uh, flagella. But if we remove the Nexin, then they will just slide past each other because there will be nothing to hold them together. This is important in terms of arrangement. In case of IFT complex B, the whole setup, which has kinesin 2 motor actually bound to the microtubule and cytoplasmic dynein piggybanking on that with the cargo, actually goes towards the plus end or the anterograde transport, while the IFT complex A is involved with the retrograde transport going towards the minus end, where the dynein is the one which is bound to the microtubule, while kinesin is the one which is piggybanking along with the adapter. Very general questions. CDKs, PIKs initiate centrosome duplication. CDKs initiate centrosome splitting in case of mitosis. Cohesin is the one which keeps them connected till the metaphase, because in anaphase they separates. So till metaphase, these sister chromatids are connected together with the help of cohesin. Okay. In prophase, two of the proteins are involved, plus tip and kinesin, not protein but molecules. Okay. Yeah. Anaphase has two distinct phases, anaphase A and anaphase B. Anaphase A is actually movement of chromosome to poles. Anaphase B involves with spindle pole separation. So anaphase A is basically pulling of these chromosomes towards the poles, and then anaphase B involves moving these poles away from each other towards the end. Okay. Also, telophase is the one where the nucleosome or nucleus, basically not nucleosome but nuclear envelope, reassembles. And here, if you see prometaphase, the nucleus dissolves, or nuclear membrane dissolves rather. Different kinds of uh, or different classes of microtubules: astral, which gives polarity; kinetochore empty, which is bound to the sister chromatids; and polar empties, which also gives polarity and basically slide past each other with the help of kinesin motors. These are, remember, anti-parallel microtubules in case of polar empty movements. Okay, so here we can see that although kinesin 13 activity, which is involved with depolymerization of the microtubule, is constant throughout, actually the XMAP 215 activity, which is basically an example, a specific example of MAP proteins or microtubule associated proteins, helps in the polymerization or stabilization of plus end. So XMAP 215 activity is high during the interphase but low during the mitosis. Why? Because of inactivation by phosphorylation. So XMAP 215 gets phosphorylated here. That is why the activity lowers. That is why the kinesin 13 activity, which was constant, appears to be high. That means more of depolymerization of the microtubule happens. That is why this is unstable condition. And during the interphase, these are stable conditions. Association: microtubules or the spindle fibers actually bound to the plus side of the kinetochore. This is the plus end or the outer layer. This is the inner layer. So the plus end of the microtubules are bound to the outer layer of the kinetochore. And this is the centromere DNA, which is part of the sister chromatid itself. Okay, very, very important, because kinesin 4 and kinesin 13, these are involved with the depolymerization here, as well as shrinkage at the plus end. On the other end, where the growth is happening, that means this microtubule is basically pushing these sister chromatids towards this middle portion. In this case, kinesin 7 is involved, excuse me, which helps in the stabilization of the plus end of the microtubule. Also, in the shrinkage side, as kinesin 4 and kinesin 13 are involved, also the dynein-dynactin activity basically takes this whole cyst chromatids towards the minus end. So both of these activities are working towards this end. And here, these sister chromatids are being pushed by the growth of this plus end by stabilization with the help of kinesin 7. Okay. Here, we just saw the structure that NDC-AT complex basically forms a sleeve-like structure and links the inner kinetochore with the plus end of the microtubule. And in this case, we discussed about what happens when these sister chromatids are under no tension versus tension. So when there is no tension, there is phosphorylation and phosphorylation of the NDC-ATs. Okay, with the help of kinase aurora B, which is present in the inner kinetochore. Okay. In case of when the sister chromatids are under tension because they are being pulled by the microtubules, what happens? The phosphatase PP1, which is associated with the outer kinetochore here, in this case, dephosphorylate the NDC-AT, and NDC-AT gets pulled apart, cannot phosphorylate. And NDC-AT binds microtubules very strongly. Okay. Here, NDC-AT is loosely bound to microtubule. So that's pretty much it about the exam review. So I'll put up an announcement very soon. The exams will have the similar format. That means sets of multiple choice and sets of true and false. I don't want to change the format right now. I was suggested to give short answer questions which involves thought processes or thinking basically, but it'll make it harder for you, frankly speaking. Uh, the fill in the blanks, not fill in the blanks, the multiple choice and true and false is a easier version of the exam, but I would prefer it this time because this is the first time we are having this online transition. Maybe in the future semester, we can have short answer or very short answer questions. So this time we'll just keep it just like the similar format as exam 3. Important points are the increase in time. So some of you have discussed about this issue that they would need their slow reader and would need more time. So I have decided to increase the time for 50 questions from 50 minutes to 75 minutes. That's the whole class time, basically 1 hour 15 minutes. That's our regular class time. The exam will start exactly at 11:30 a.m. on Wednesday, this Wednesday, and will go on for 75 minutes for pretty much everyone except the OSS students who will get 1.5x extra time. And what else? Some A few have mentioned that they have something important which is ending at 11 or at 11:30. So even if you take the exam 10 or 15 minutes later, you will still get 75 minutes of time. So don't worry about that. But I would prefer that everybody starts at the same time. Also, this is taken from a pool of questions and will be randomized. So mostly you won't get same question. Okay. About the review, there won't be any review as have been mentioned during the first class of this section itself. So I mentioned that there won't be any review for the final exam. Uh, you might not see your scores on e-learning immediately after the exam, but it will be shown on e-learning by the weekend. Okay. About the curve, I'll have to compile all the data and then I'll have to discuss with Dr. Shrikant, who taught the first half. And then we have to decide based on the overall class performance whether you need a curve and what kind of a range for different grades. We'll decide then. So I cannot say anything right now. So when it will happen, we'll just mention that to you through announcements and you'll get to know automatically. So please do not email about uh, what will be the curve and all. You will get to know eventually. If you have any specific queries, you are always welcome to email. Also, you still have a couple of days in between. So study well, focus on the material. Do not depend solely on this review alone. This review will only be helpful if you have gone over all the lecture recordings, gone over all the slides carefully, and have understood and clarified any doubts. And then when you hear this review, it is going to be helpful. But if someone thinks that I'll just listen to this review and I should be all right, probably not. Okay. So wish you all the best and all the best for the future. We are not going to meet in person. I'm not sure whether that'll happen in the future or not, but my good wishes are always with you. Do well in life. Uh, try not to give excuses whenever you work, wherever you work in the future. Also, be confident and just do your best. Like if you can give 100%, try to give 105%. Okay. And when you focus on yourself rather than others' faults, you will improve better. Because as individual person, we can only work on ourselves. Okay. So focus on your strengths, focus on your plus points, and keep on improving them. Keep on, or at least keep on trying to become a better person compared to your previous personality the previous day. So each day try to become a better version of yourself. Okay. So with all this, again, all the best for the future, all the best for the exam, and I'll stop here. All the best.